Thank you very much. Thank you very much for inviting me. It's really a pleasure to be here, and thank you for hosting us in this really beautiful city. Okay, so because this is an educational day, I first wanted to kind of give you this really exciting road to single EV imaging. I think we all appreciate the role EVs have in intracellular communication. We are really excited because they're potential biomarker sources, they're potential therapeutic delivery vectors, but we still need to characterize them. And ideally, we really want to get a lot of parameters about these vesicles. We want to look at their size, we want to look at their cargo, we want to look at their shape, right? So where we started and how did we get here? So we started in 1500s because that's really where the fluorescence was discovered in the bark of the Mexican tree. And then there was this kind of really long period. You see these beautiful images of cork cells that were drawn by Hooke. And then, somewhere around 1940s, Kunz got these beautiful images of cells done by immunofluorescence. Only in 1967, we started to see this really excited platelet dust. So we are starting to see EVs with EM. And then it took a couple more decades before we were able to see these lovely vesicles with fluorescence microscopy. And this is really where I will be focusing today. So how do we see these vesicles with fluorescence microscopy and why this is useful and what it can tell us? And along the way, some of the mistakes we can make and some of the lessons we learned. So how do we label vesicles, right? So we can covalently label extracellular vesicles using either maleimide dyes or NHS esters, right? So we need to have a dye, and then we can conjugate it to this kind of outer layer of vesicles. We can use genetic labeling. This is a little harder when we consider vesicles from blood. We can do affinity labeling. You've seen a lot of talks today that dealt with this. So you can have aptamers, you can have antibodies, you can have all sorts of different affinity reagents. But this is really crucial. We really have to be careful that we are looking at specific reagents that have good binding capacity. And also, we can use these newly developed and some of the older but established dyes that will really interculate into the membrane and label the membrane. So these are some of the ways we can label vesicles. But how can we still image individual EVs? So many of us are using confocal microscopy and wide field microscopy. These are conventional fluorescence microscopy techniques, but they're limited to about 250 nanometers. And about 20 years ago, we started developing these really exciting super resolution microscopy methods that can see very small things apart. And their resolution can now go to the angstrom level. So it's really exciting to see where this technology is going. And why do we care, right? Well, extracellular vesicles have this really wide range of sizes. They can go down to 30 nanometers and then they go to 10 microns, right? If you look at the oncosomes. So we have this inherent heterogeneity in these vesicles. So we need to find ways and techniques that can describe them well. So SMLM, or single molecule localization microscopy, is ideal for that particular tasks. So what we did in our lab, we wanted to use the benefits of SMLM and apply it to EVs. As we heard today, EVs are exciting, but EVs are in a mixture inside of the blood. There are lipoproteins, all aggregates, many different components, right? And on top of it, often we care about specific type of EVs, right? We care about EVs from cancer cells, or we care about EVs from specific organs. So we really need to find ways how to fish out those that we care about. So what we really did in the lab, Anders led this 7 methodology, single extracellular vesicle nanoscopy, that effectively combines affinity isolation of extracellular vesicles and single molecule molecule localization microscopy. And this is kind of a mix and match technique. So we make our own chips, so they're really cheap. They're, a couple of dollars per chip. And we can really decide what we want to isolate and what we want to stain. So this is really nice because we want to make this available to everyone. And here I'm showing you tetraspanins. We just heard a beautiful talk that says that tetraspanin CD9, CD81, and CD63 are common on EVs. So often I will be looking at these T-span enriched EVs, and that's what we use when we start developing the techniques. And then we image and we do a lot of data analysis. Okay, so what we will learn along the way? Well, samples have platelets often. So we, because this is a kind of a nice affinity based method, so if samples do have platelets, we actually see them like in this image here. So we are very sensitive to that, and we kind of have built-in technology to at least see if samples do have platelets. So please, double spin always helps. Another thing is aggregates. Right? So there are different isolation methods. There is nothing perfect, but some can really clump up those EVs. And we tend to see that. So if you see that there is a problem with your prep, don't go further because you're not going to get anything meaningful. So, getting these pre-analytical methods right really makes a difference. And then buffers, right? So we heard about all these lovely methods that help us isolate EVs. And these are recently published papers by Samir Al-Andalusi. And you can see that there are buffers that will make EVs be more stable when they're stored, and they will help you isolate higher quantities of EVs. So we have to be thinking about these methods. Now, can I always have albumin in my buffer? No, right? If you're doing mass spectrometry, that's really hard because we have to worry about dynamic range. But if you're doing microscopy, then it's perfectly fine. So those are all the things that we kind of have to weigh as we are going forward in this particular direction. And then all the other things that we need to report, right? Biological properties. These are data from rat samples that we done in the lab. And we've seen a huge difference between females and males. We don't typically observe this as much in other samples, but with rats, we've seen it. So we do need to think about these properties, biological properties of samples that we are investigating. Okay. And here is the very important lesson that I learned. There is no best method. There is the optimal method for what I'm trying to answer, right? So this is NanoView image, a beautiful image from Randy Carney's lab. And NanoView can isolate EVs. We can see if markers are there or not. It can measure size by interferometry. But it's much faster than what we are doing with our imaging, right? Perhaps you have an instrument in your lab or in your institute or not, right? That's the biggest consideration right now. Or what is the price per chip? Sometimes things are expensive, right? So we actually have to consider many things as we go along and then decide what works for us. And here is kind of an example where we compared ExoView and 7. What are the things that we care about? Well, we actually do have to care about, capturing efficiency. Do we capture enough EVs? Because that means, are we sampling well? And, two methods do pretty well, right? And then we need to care about sizes, right? What is the size range of a particular instrument? Interferometry has a little bit narrower size range, but maybe that's good enough, right, for some experiments. Whereas SMLM has this beautiful size range, and sometimes that's necessary. So these are all considerations. And then finally, do we really need to molecularly count, or we actually care is marker present or absent, right? So those are all considerations as we are going forward with our experiments. And this is something that I wanted to point out, methods that we use to isolate EVs matter, right? So these are experiments that we obtained using 7, and we looked at plasma EVs that we either isolated directly or we use size exclusion chromatography. And there is no question, size exclusion chromatography gets you these beautiful pure EVs, largely pure EVs, right? It's really nice, but right away you will notice that you lose a lot, right? So this is about 4-fold loss that we observe using SEC. And, sometimes I can afford that, but sometimes I cannot. So those are again, considerations if you're thinking about which experiments are you going to do. So both perfectly linear, right? You have beautiful signal to noise, but can you afford it? But this is something that I'm actually really excited to think about. And that is, what kind of properties are those EVs having? Are they identical if you look at something that comes from SEC or from plasma? And sizes are not much different, but what's really different is number of detected tetraspanins. So it's really likely we are hearing about corona and beautiful work that Edith has. SEC can remove those loosely associated corona proteins, and now you have more spaces for antibodies to bind. So we are talking about detected molecules, right, in both cases. And if you have abundant corona proteins, you can detect fewer molecules. So these are all, again, considerations for us as we are looking at our experiments. And is that good or bad? Well, it's a question for biomarkers. Sometimes these corona proteins are really relevant, and sometimes they're, making our lives much harder. Okay, so I need to get more EVs on my samples. We are always in a rush to get more EVs. So let's look at other methods. And this is exactly what we recently did. We looked at Captacor resin, 700 and 400, PEG precipitation, thrombin treatment, and just pure plasma dilution. And what you obviously know, I didn't even need to show you the gel, these are not going to be as pure, right, as SEC. So you're compromising, but again, for imaging, that is okay. What really wanted to point out that the Captacor 700 resin does follow those little EVs. Right, so you have fewer EVs, and those are generally larger in size, and that really tells me that these pores of the Captacor 700 resin are actually pretty large enough, and those small EVs can disappear. So that's why it's important to consider all of these things going forward. Okay, that's great. These are tetraspanin enriched EVs, but most of us care about those rare EVs, right? So this is an example of HER2 enriched EVs from plasma, and this is work of Nanjiang in my lab that was recently done. So we compared all these lovely different methods for isolation again, and wanted to see how they look like. And then we looked how many EVs can we get. So this is a HER2 positive breast cancer patient. So we wanted someone who has an advanced disease. Can we count as many EVs as we can? And thrombin treatment really gave me the most HER2 enriched EVs. And the reason why this is happening is because this is where I can really fit the most plasma on my samples, right? Other methods I have to dilute and compromise there. Plasma, you can't just put it on the cover slip as is. It's too dense. Your affinity reagents will not bind because they're occluded by all the other particles that are present there. So you have to go at least 1 to 6 dilution. So thrombin treatment works really great here. And then we also wanted to check, is it working similarly in a control? So we had the triple negative breast cancer patient and healthy control. So very few EVs there, so that was good. So we figured out that thrombin treatment works for her too. But I really want to make sure that this is not one size fits all. We had several other projects in the lab, and thrombin treatment wasn't best for all of them. And that is logical, right? You actually have to think of what you're trying to detect, what population you care about. Okay, so what Nan did, she took patients that either had HER2 enhanced tumors, so they had higher expression of HER2. We looked at triple negative patients, and we also had a population of patients that were originally diagnosed with triple negative cancer, but at the time of blood collection, the cancer was not there. And we had 4 patients per group, and we looked at many regions of interest through ROI. And what we really noticed right away is that these patients that have elevated HER2 have vesicles that also have a lot of tetraspanin. So you can see that there is pretty big difference in tetraspanin content. They're generally even a little larger. So what we really did here is we did gating. So we know that the tetraspanin content is different. We know that the size is different. So we don't only care, are those EVs positive for HER2, but we care about their properties. So we generated this 2D histogram. Each dot on this histogram is an EV, and for each EV, we will give you number of detected tetraspanin and size, and we'll gate them. We will gate them exactly like you would do in flow, and when we gate them, we can really nicely distinguish those that are from patients that have elevated HER2 levels and those that have triple negative breast cancer. So that was really nice. And you can even see that we can detect patients that have this IHC1. This is immunohistochemistry level 1 that is just slightly elevated level. So this was really nice to see. But, we all know that EVs are very diverse, so I love these bubbles flowing out of them. We are still learning about them, and we have these newest suprameres and exomeres, and we don't really even know, exactly how they originate. But can we detect those smaller things from plasma? And this is because plasma is so complex. We wanted to see, can we just get those non-vesicular extracellular particles? Because 7 has the beautiful resolution of about 7 nanometers. So, you can start seeing stuff that is really small, such as like 15 nanometers or so. And indeed, we were able to see this population that is significantly smaller in size, but you can also see it's more elongated. So the circularity tells you something about shape. So something that is perfect circle is 1, and the longer it kind of elongates, you will see the less circular it is, the number drops down. So you can see really nicely how we can separate these two different levels. But what's also really nice is that we can confirm this in isolation method. So this is collaboration with Soumya Das, and they isolated suprameres, and we were actually able to see this kind of same pattern. So, we are still learning about these particles, so we also need to develop method to image them and analyze them. And then I will just finish with this kind of a thought experiment, right? So we all love single EV imaging. It can bring a lot of good, but it requires often special equipment. And I really loved what Andreas was saying. So can we do something more simple, right? So, and can we do something that everyone can use? So this is kind of my attempt of thinking about democratizing single EV imaging. So Henrique's lab has developed this really wonderful super-resolution imaging method called super-resolution radial fluctuations microscopy. So you acquire just a short sequence of images, and then you can correlate them in the radial dimension and temporal dimension, and basically the signal will correlate and noise will not. So you can see that we started from EV images on the TERF side, and it's still blurry, you can still see them. And on the right side, they are nice and crisp and lovely, right? So it works really nicely, but we still have to analyze these images. And this is really where Anders was kind of leading this effort to do that. And we spend a lot of time in a microscope room. So we actually have a lot of time to come up with acronyms. So this was 7UP. This was 7 Universal Protocol. And this is really a machine learning based method where we kind of did SMLM and SURF side by side. And we basically trained our method to segment vesicles. So you can see these beautiful segments on this image. These are plasma EVs, and you can see that, red dots, those are still SMLM. So you can see the differences, right? SMLM is going to be smaller and nicer. So you can see a few of the images enlarged here and then a few there. And you can see that we can really resolve these big blobs that are actually not looking separated by eye. So this method works really nicely. And I wanted to show you a confusion matrix. So you can actually see that when we have a single segment, most of our EVs, around 10,000, we do really well in our tests. We do really well with 2 segments, slightly worse in 3 and 4 segments. But remember that our machine learning model had very few those multiple segments, so this was really expected. So the method works really nicely. And we still need to look at the sizes, the molecular count, so we can do really lovely correlations. And this is our training set. These are recombinant EVs for Ann Hendricks's lab that are commercially available. We like to use standards throughout. I didn't mention much of that, but we kind of keep calibrating instrument, keep using standards throughout. So we can see beautiful correlation in sizes. And then if we image on wide field microscope or turf microscope, we get really comparable sizes. So this is the beauty of this. We want to make sure that everyone can do this. And then we can do the similar correlation with molecular counts. These are the tetraspanning counts. Works really nicely. Again, there is no difference between SMLM on average and with wide field and turf. But, can we distinguish between two different types of EVs? And indeed we can. These are plasma EVs that I just showed you, and they're slightly smaller than these recombinant EVs, and indeed we see them as slightly smaller than recombinant EVs. So this was really exciting, and we really look forward to making this further for use of others. So I hope that I showed you today that single EV imaging with fluorescence microscopy can be really useful for characterization. There are many parameters that we need to consider. We love that these EVs have heterogeneity. We need to find ways to describe them, right? We can look at the membrane, we can look at the cargo, we can look at the shape. Those are all important properties, and we should not forget about them as we are thinking about our EVs having a marker or not. And finally, I would really like to acknowledge many people who did the work. So Andras and Balint really did surf imaging and made it as a 7-up. And Anders did 7, Nan did a lot of HER2 work, and Sarah, Ben, and Jonathan did quite a bit along the way. So I'm really grateful for the funding sources, and thank you very much for your attention.
Okay, there's time for a few questions.
Just one question from the chat. Can we get 7 done for our samples at your lab?
So actually, you can do it on any super resolution microscope, right? So that's kind of the ONI microscope, Abelite, Nikon. I am agnostics when it comes to the microscope. So we have the really protocol that is super detailed, so anyone can spot any set of EVs that they wanted to do. So, obviously we are there to help, but, hopefully this is useful.
Excellent.
Yeah.
Sorry, microphone.
They will prevent you.
Okay,
Yeah.
they will prevent me, okay. So what's the thrombin method?
Oh, so we basically just defibrinate the plasma. We add the thrombin and it reduces the viscosity. We spin it down, and that's the only thing we do. So it's a simple addition. So like ExoQuick, right? Sometimes you pre-clear it with thrombin. So it's very similar way of doing it. It reduces viscosity, allows EVs to bind. And I absolutely agree with you. I was very worried about it. That's why I kind of put this whole set of comments, right? It may not work for everything, but as many times as we tested it with HER2, it worked beautifully, right? So it may not be a universal thing. It allowed us to get the most plasma that we could, and that's why it was useful right here.
Youngin Park from Korea. Great talk. I have a question. So can 7 system visualize the double or triple layering of the EV? So we have been seeing the double or triple layered EVs from EM, whether... Yeah.
So that's a great question. So the resolution that we get right now is about 7 nanometers. So if we have a way, so if some of those beautiful EM images show pretty good separation, right? So if we have a ways to label them, we would be able to see the double layers, right? So as long as you have a way to label this inner membrane, you would be able to do it if they're separated for more than, 7 nanometers, but 2 times sigma, I always worry about 2 times sigma as well, in those things. But for some of them, I would say yes.